Good afternoon, everyone, and thank you for attending this session of Grand Rounds. I'd like to welcome our presenter for today, Dr. Alan Junkins, who will be presenting on the topic of gram-negatives in the blood. At this time, I'm going to stop sharing my screen and turn the presentation over to Dr. Junkins.
Okay, hey, everybody. Let me get this up and going. Okay, I think we're good. Everybody can see that?
Looks great.
Okay, yeah, today we're going to talk about, we've already talked about blood cultures before, but today is kind of specific about gram-negatives in blood cultures. And the reason I want to cover those, why gram-negatives, and this is my personal opinion. I've always been a gram-negative person. I've always just felt like they are cool. I don't want to downplay the excellence of Staph aureus. I mean, he's a really good pathogen, or strep pneumo, or group A strep, really one of the best pathogens out there, group A strep. But I'm just find the gram-negatives a little bit more interesting as a whole. And some of the reasons, I'll tell you some of the reasons why I like this. One of them is less predictable resistance patterns. If you've got a Staph aureus and you got a MEK-A gene in there, you know what that is. A MEK-A behaves the same all the time. You know you're going to be resistant to all those beta-lactams, and you know great majority of the time you're going to maintain susceptibility to a few of the newer drugs. Same thing with Enterococcus with the MAN-A gene. Those genes are very straightforward. They do what they do. But when you get the gram-negative rods, you've got this whole slew of resistance genes. And just because you've got one ESBL gene, you don't know exactly what that's going to mean as far as the spectrum of activity, what drugs it will work against and which ones it won't. And they differ so much in how they express those. Sometimes they turn it on, sometimes they leave it silent. So it's just a lot less predictable when you come to gram-negatives. I also look at the clinical significance in blood cultures, for example. I mean, when we talk about contaminants in blood cultures, we're always talking about gram-positive organisms. The whole list of potential potential contaminants are gram-positives. What contaminants can you think of? What organism can you think of that are commonly contaminants in blood cultures that are gram-negative? And there just aren't any. I'm not saying that gram-negatives don't contaminate blood cultures. Of course they do. But when you get a gram-negative in a blood culture, the default is that this is going to be a clinically significant organism. Let me give you an example here. This is a paper that came out in 1997, so it's old, and it's actually based on data from 1992 and 1993, so it's really 30 years old now. If I did my math right there. But nothing's really changed a whole lot as far as this goes. What these people did is they took a whole bunch of blood cultures and they looked at every single isolate that came from those blood cultures and really did an in-depth study, looked at that patient's chart, everything that was on with that patient, and decided whether this was a contaminant or not, or whether it was indeterminate, whether they couldn't tell. So you got gram-positives on the left here, and it kind of shows you how many times. Now look, Corynebacterium, they found 1 out of 53 that they thought may have been real. The other 52 out of 53 were definite contaminants. Cutibacterium, every single one was a definite contaminant. We all know coagulative staph, 82% of the time they say it was a definite contaminant. If you look more at the data, it's like 10, 12% of the time it's questionable. So even some things like Enterococcus, group B strep, that's only 15 isolates, but there was some contaminants there. Then on the right side, you look at the gram-negatives. And first of all, you can notice here that there's a lot fewer gram-negatives in blood cultures than there are gram-positives. And that's largely because the gram-positives are going to be colonizing the skin more than the gram-negatives do. So they are going to be found more often in those blood cultures. But I mean, look at this. I mean, one Acinetobacter baumannii, can't even say that. Baumannii that found that was considered a contaminant, 1 out of 16. Even got a single isolate of Pseudomonas aeruginosa, they decided it was a contaminant. Those are both environmental organisms. It makes sense that they would contaminate every now and then. But all the others, no contaminants. They are all clinically significant. So when you get a gram-negative in a blood culture, you know it's going to be something important to deal with. The other reason that makes them interesting is that gram-negatives in general, on the whole, will induce a more potent inflammatory response than gram-positives do. All gram-negatives have in their outer membrane this thing called lipopolysaccharide, which is diagrammed over on the right there. Lipopolysaccharide. It's the only place in nature you're going to find lipopolysaccharide. You might find it contaminating some solutions at some point. There are tests where they would test in pharmacy where they'll test certain solutions to see whether it has lipopolysaccharide or endotoxin in it. But that came from a gram-negative organism, just doesn't exist anywhere else in nature. LPS is catnip for white blood cells. There is nothing else that drives them crazy like lipopolysaccharide. When they see it, they know what they've got. And you can kind of see from this diagram here, the bacteria, this is an integral part of their outer membrane. They can't just blow it off, lose it, like some bacterial toxins do. But what they do is they release these outer membrane vesicles, these little blebs, they're called micelles as well, that are just little balls made by this outer membrane. And it contains lipopolysaccharide, and that goes off and stimulates these white blood cells. Release of cytokines, complement cascade, coagulation cascades. It's crazy how complicated it is. I would love to be able to explain it to you. And even if I had 2 hours to sit here, I could not explain it to you because it's a very complicated. There are entire journals that are dedicated to the mechanism by which endotoxin or lipopolysaccharide works. They have entire meetings. They all get together every summer and they talk about how endotoxin works. It's very, very complicated. But let's just say that white blood cells go nuts. And so I found this. This is the extent that I can explain it to you, which is exactly what I just said. They release these little blebs of this lipopolysaccharide. It, Zorro there, activates white blood cells, and they release all kinds of inflammatory mediators, which leads to inflammation, which leads to activation of white blood cells some more. So you're going to be more likely to get a strong inflammatory response and sepsis from a gram-negative than you would a gram-positive. Now, like I said, I'm not kind of bad-mouthing group A strep or Staph aureus because they produce other things that can cause major problems. But the typical gram-negative is going to give you more inflammation than typical gram-positive. This is a study, another old study, a lot of these are old studies, 2010, where these people looked at people who had 3 different degrees of sepsis, what they call just standard sepsis, those who had severe sepsis, and those who had septic shock. And then they looked to see whether they had gram-positives or gram-negatives in their bloodstream. Now, the way they show this here is that you can see if you have gram-positive bacteremia, you tend to have what I could call standard sepsis, 72% of them. So it moved to the left of the scale here, the lower end of the scale. Whereas if you had gram-negative bacteremia, you're moving up to the higher end of the scale. 44% of them had full-blown septic shock. So they're looking at that and saying that's statistically significant. The distribution between the 3 different grades of sepsis. I kind of want to look at the other way. So I kind of took their statistics and turned it around. And I probably broke some statistical rules in doing this, but we're going to do it anyway. Because I would look at it and say, if you have gram-positive bacteremia, if you're in this group here, if you're in one of these groupings here and have gram-positive bacteremia, there's a 15% chance that you're going to be up here and have septic shock. That's 25 divided by 168. Okay. But if you have gram-negative bacteremia, then there's a 36% chance, more than twice as likely that you're going to go right into septic shock. So that, again, is a p-value right there in the same ballpark as what they found for that. So gram-negative bacteremia is more likely to get you into that high, more severe range. This is from the same paper here, and it's not a very exciting table. I just thought I would show it to you anyway. All these different characteristics between people who had gram-positives in this column, people who had gram negatives in this column. And over here, they had some people who had both a gram positive and a gram negative. The only thing that was significant was the Apache score. So this is used to grade the severity of a person's illness. So the people who had gram negatives were more severely ill than those who had gram positives. To me, I look at the mortality and say, if you have gram negative, there's a 40% chance of mortality as opposed to a 28% chance of mortality. I would prefer not to have that extra 12% chance of mortality if it were me. It is kind of interesting that if you have both a gram positive and a gram negative, then you're actually in between. So it's kind of like if you've got E. coli bacteremia, you're in bad shape. But if you have E. coli and viridans strep at the same time, you're going to be okay. It's kind of interesting, but So again, gram-negative bacteremia is going to make you sicker and more likely to have a bad outcome. So I'm going to show you some of our local data. This is the year 2022, all the gram-negatives that we got out of blood cultures and the quantities of them here at Norton Healthcare. So you can see the leader by a long shot is E. coli, is kind of be expected. Klebpneumo number 2, Pseudomonas aeruginosa number 3, and then we kind of run down with a lot more of these Enterobacteriales organisms. Bacteroides fragilis is our biggest anaerobe organism, a common gut organism there. And then you move on down. Haemophilus influenza being an important one from the upper respiratory tract, and you move on down from there. One thing that's really interesting for me is there's no Acinetobacter on this list. We will talk about Acinetobacter here in a bit. It's a bug of great clinical interest and epidemiological interest, but we just didn't get much from blood cultures in 2022. It's higher up on most laboratories' list. Now, we have talked before in a couple of presentations about the rapid detection of organisms or resistance markers in blood cultures and positive blood cultures. Now, I wanted to share this. Here, we use the Verigene, and all these little smiley faces show you the bugs that the Verigene is capable of detecting. So we would find out after the blood culture comes positive, we would do the gram stain, and then we could set up the Verigene, and a couple hours later, we would be able to know whether it was one of these organisms. There's others. The BioFire BCID2 is the most popular one used around the country. And then there's the GenMark ePlex, which is a really cool test. To my knowledge, not as commonly used as the other two. So you can see who's going to get picked up. So for us, the most common organism that Verigene does not pick up from the gram-negative side is going to be Serratia marcescens. And you do see that quite a bit. That's an environmental organism. Usually, you see it commonly in people who have, well, what do they have? They have endocarditis. It's fairly common in people who use IV drugs, that type of thing. So from our perspective, we look at this and say, of these ones that the Verigene detects, only about 80% of the bugs that we get from gram-negative bacteremia is the Verigene able to detect. So about 20% of the time, it's not going to detect the organism. BCID and GenMark are going to be a little higher than that. But notice the gram-positives are also higher than that. If I did this exact same table with gram-positives, you'd see that about 88% of our gram-positive isolates would be picked up by the Verigene. So what I'm saying is that with gram-negatives, and it would be the same thing if you use one of the other two as well. With gram-negatives, you're going to have a slightly higher number of organisms that are not detected. And so you don't get that rapid identification, and you have to wait for the culture results to see. Where do these gram-negatives come from? There's two main places. There's various body sites. So you know good and well that these enteric organisms like E. coli and Klebpneumo and the obligate anaerobes like Bacteroides are long-term colonizers of the intestinal tract. They live down there in massive quantities in your colon. And almost all of the organisms, even some of the environmental bugs, are transient colonizers. You can certainly get some of these other organisms living in your colon for a short period of time. The upper respiratory tract, I look at most of these. You have bugs like Haemophilus influenza and some other gram-negatives, Neisseria meningitidis and normal flora Neisseria, for example, that are long-term colonizers of the upper respiratory tract. Most of the bugs that I think of, like E. coli and Klebnumo, Citrobacter, Serratia, those will be what I would call more likely transient colonization of the upper respiratory tract. They don't get a good foothold there. They will stop in for a while, stay for a while, but then they're not gonna get massive quantities. The exception there is when people are sick. There was a really cool paper that was done probably 10, 15 years ago now, which showed that people who are ill, and it doesn't really matter what it is you're ill from. You could be ill from COVID, you can be ill because you had a stroke. But when you have this general illness in your body, it causes additional fibronectin receptors to be exposed in your upper respiratory tract, which makes it easier for gram-negatives to colonize. So that's why we tend to see people who are hospitalized, who are sick, end up more likely to be colonized in the upper respiratory tract with gram-negatives. And of course, you got to look at your pets as well, because they've got it in their poop too. Now, some of these gram-negatives are environmental organisms. I look at bugs like Pseudomonas and Acinetobacter. They are primarily bugs that live in soil. They're in dirt, they're in dust, and therefore they're on any surfaces that are contaminated with those with soil or dust. Serratia largely is the same thing as well. That's really not a bug that colonizes the intestinal tract to any great extent. If it's in the environment, it's going to end up in water. Everything gets washed into the water. And so some of these bugs are really, really good. Pseudomonas is really, really good at surviving in water, living off whatever organic material it can find in the water. So water is always going to end up being a problem with these environmental organisms. And of course, anything that's fecally contaminated, sewage plants are going to be loaded with intestinal flora. These bugs most of these bugs tend to survive in the environment pretty well. They're fairly hardy. You know, a strep pneumo is going to die pretty quickly in the environment. An E. coli is not. They're going to be a little tougher to deal with. When we talk about bacteremia, we'll often talk about primary bacteremia, which means this is the first thing that's showing up. It's the primary disease we're seeing. It's showing up. There's no evident source of where this bug came from. Whereas a secondary bacteremia will be there's an infection someplace else, or there's colonization someplace else. There's a patient who's got pneumonia, that organism, say Klebnumo, gets from that pneumonia into the bloodstream. Or there's a urinary tract infection with E. coli, that E. coli gets into the bloodstream. Those are secondary bacteremias. Now you can look at this and say, what that means basically is that primary is kind of like throwing up the white flag. All of these have a source. All of these came from somewhere. The fact that you don't know what it is, you end up calling it primary bacteremia, but it was from someplace. So, and this is just a cool little thing. I thought this was pretty neat. You know, some guy standing here. This paper here looked at what they consider to be the top four, the big four gram-negative bacteremia. And we've already seen with our numbers, E. coli, Klebnumo, and Pseudomonas aeruginosa are the top three in numbers. Acinetobacter, for us, was not in the top 20. Okay, it was way down the list. Others have said Acinetobacter is probably somewhere in the top 10, maybe like number 6, 7, 8 for a lot of places. But even if it's not high up there in numbers, it's important because of its drug resistance capabilities. It can develop resistances very, very easily. So they made this cute little graph, which I thought was kind of fun. E. coli hangs out right here, GI tract and the urinary tract. Klebnumo likes the gastrointestinal tract and the lungs. Pseudomonas aeruginosa can colonize the lungs and the skin. And Acinetobacter does the skin and the urine. Now, there's a little crossover here. Acinetobacter likes to go to the lungs as well, especially, you know, you've seen these patients with this hospital-acquired pneumonias, vent-associated pneumonias. Acinetobacter shows up in that a lot. And Klebnumo is a common urinary tract infection bug as well. So these bugs tend to get in those sites and then somehow gain access into the bloodstream. So Let's go through the process. We've already talked about blood cultures, and I'm certainly not going to talk about blood culture collection today, but we've talked about that. But step one in diagnosing these gram-negative bacteremias is going to be, of course, blood culture collection. That blood culture is still the way to do it. So we collect the blood. Then we're going to incubate the blood. We're going to take the bottle and we're going to put it directly on an instrument that's going to do the incubation for us. These are the three main types. This over here on the left is the BactiAlert Virtuo from bioMérieux. Very cool instrument, really the state-of-the-art instrument that we have available right now. This one in the middle is the Versatrek, which is number 3 on the list, has a far lower share of the market. And this is the BDFX on the right here. This is what we currently use in our laboratory. All of these are pretty much the same thing. We're going to take that bottle, we're going to put it on that machine. That machine is going to detect gas production, CO2 production from metabolism of those organisms that are in the blood culture, if there's organisms in there. And then they will flag us, their lab staff, when there is evidence that there is a bug growing in that bottle. So in the case of the FX that we have, this red light will come on and it'll make this little pinging sound, and we'll know, go over there right now and pull that positive blood culture off. You really shouldn't get this picture. This is kind of a fake picture. You shouldn't get all those lights on at the same time, but you know, it makes for a pretty picture. So when the blood comes off, the next thing we do is a gram stain. You would think blood culture gram stains should be fairly easy to interpret, because there's, generally speaking, only going to be one bug growing under. We don't get, well, we do, but it's very rare for us to get blood cultures that have 3, 4, 5 bugs in there. That's contamination of some sort. It's okay, you know, maybe about 10% of the time we'll get a blood culture that's got 2 bugs in it. But it should be fairly easy to tell. Let me tell you first off, though, that these 3 pictures that you're seeing here are not from blood cultures, okay? So these are really straightforward pictures here, just from a colony off a plate. Over here on the left, we have an E. coli. And that's exactly what a standard gram-negative rod is going to look like. You can see it's definitely a rod. It's definitely longer than it is wide, okay? But it's not too incredibly long. That's kind of a standard, you know, 1 micron across, 4 microns long, that kind of thing. That's a standard gram-negative rod. I can't look at that and tell you it's E. coli. I just happen to know because I put the picture in here. Almost all your gram-negative rods can look very much like that. And so when we give you a gram stain report, we're going to say gram-negative rods or gram-negative bacilli. We can't even give you any hints most of the time, kind of like with gram-positives, where we say gram-positive cocci pairs and clusters versus pairs and chains. Can't really make that distinction with gram-negatives. This one in the middle, if you look at that, you look at it and those are smaller. They're still definitely rods. Some of them are definitely rods, but they're smaller, they're shorter. So maybe they're 1 micron across and only 2 microns long. Coccobacilli kind of look. That's a Haemophilus influenza. I'll be honest with you, if we saw that, I really wouldn't want to suggest to you, oh, no, don't worry, I think it's Haemophilus influenza. Because the length of the bug depends on its growth phase. You will see short little E. coli sometimes. So you have to be kind of careful. We can look at that and say, you know, I think that looks like Haemophilus. But I'm not going to tell you that. I'm not going to put that in the report. Don't worry, it's Haemophilus. I shouldn't say don't worry. Haemophilus is a bad bug. The one on the right here, on the other hand, look at that. Long, skinny, very long, very skinny bugs. That's called a fusiform morphology. And when I see that, I'm thinking Fusobacterium, Capnocytophaga, Leptotrichia. Those are the 3 bugs that are going to look like that. On the other hand, I have to be careful. When we see that, we're going to report gram-negative rods. We're not really going to tell you anything else more from that because we're not too certain about what that is. So you don't get a whole lot of extra information from us other than gram-negative rods. Now, let me tell you, it's a little harder than those pictures. This is an actual blood culture gram stain that we did a couple weeks ago. And first of all, you can see all this pink background. That's all this proteinaceous material from blood. Blood has a lot of that stuff in it. And unfortunately, it's pink. And unfortunately, gram-negative rods are pink as well. So if you look at this, you can see some things that may look like bacteria. You may even look at that and say, I think that's gram-positive cocci chains right there. Or what is this? Or what is that? Here's a couple of things that look like a nice gram-negative rod right there. And then there's all these clusters of stuff, which, what is that? Is that a bunch of bugs that are clustering together? And our tech looked at this and called it gram-negative rods, and the next day it grew Pseudomonas aeruginosa and just Pseudomonas aeruginosa. So it can be a lot harder to interpret. I want to show you this one. This is one from a week or two ago too. We're talking about gram-negatives here, not necessarily gram-negative rods, right? So when we looked at this, we had a lot of questions about this. The tech who read it called it gram-negative Diplococci, which tends to freak everybody out because you're thinking, oh gosh, he's got Neisseria meningitidis, or you may be thinking this person's got Neisseria gonorrhea. When I looked at it, I said, that does look like gram-negative Diplococci, but some of them are a little big. See, this right here is a picture of Neisseria. It's not a very good picture, but you can kind of see these little Diplococci. These look a little bit on the large side to be Neisseria. This actually ended up being Acinetobacter lawfi. So this really is a gram-negative rod, not gram-negative cocci. So that threw people off for a little while there. And I want to show you this too. These are really pretty cool. I just showed you that picture of Fusobacteria and that fusiform morphology. So you could look at that and say, oh, this is one of those fusiform bugs, And this is one of those fusiform bugs over here. They're not. This right here is a salmonella. So it should look just like an E. coli, a short standard rod. These are way, way longer than they should be. And the reason is this patient was being treated with ceftriaxone when they collected the blood culture. That drug didn't kill it, but it drove him crazy. It stopped him from dividing. So he would grow, but he couldn't divide. This over here on the right side is a clebpneumo that the patient was being treated with ampicillin. Even though we technically consider clebpneumo as being completely resistant to ampicillin, it's still going to cause some cell division problem and give it these little aneurysms and stuff. So it can be really difficult when you see these in a gram stain to really tell what's going on. Okay, let me get through here real quick. After we do the gram stain, we're going to do a rapid gene detection method. Most laboratories will do this now. Here are the three: the GenMark, the BCID on the BioFire, and the Verigene. Those are your three choices. And I want to show you, this is a report from the Verigene. Verigene is what we've got, so this is what I'm able to show you. So this is what we're going to see when we get at the other end. First of all, you get a warning on this saying just because we didn't detect a resistance gene does not mean the bug is susceptible. So in this case, we didn't detect any carbapenemase genes. That does not mean that carbapenem is going to be susceptible, because there are other ways to be resistant to carbapenem. That's just the company's way of covering their, Here's the result. This is an E. coli that was detected. None of these can separate between E. coli and Shigella. E. coli and Shigella really genetically are the same species. They're so closely related. We ought to just get rid of the genus Shigella and call it all E. coli, but we're not going to do that. They put that on there. Now, to tell you the truth, in the entire years that we've been doing this, we have never gotten Shigella from a blood culture. So we're pretty confident this is actually E. coli. Here's the details of the bugs that it can detect. It did not detect any of those 7, but did detect E. coli. This is the ESBL gene, CTXM, not detected. And these are 5 carbapenemase genes, also they're not detected. Now, real quick, let me kind of hit the advantages and disadvantages and quirks of these things. First of all, none of them will report detection of a resistance gene unless it detects a compatible organism. So if, for example, Verigene does not detect Serratia marcescens, if we had a Serratia marcescens that was producing a KPC, Verigene would not give me that KPC result. So I have to have an organism detected in order to get any of the resistance genes. detect it. Serratia marcescens actually has been shown to produce all 6 of those different carbapenemases. But if we have one of those in our blood culture, we'll have no way of knowing that. We have gotten NDM producing Providentia reticuli from blood culture before, unable to detect it entirely. Second problem, it doesn't detect all the carbapenemase genes. There are some other carbapenemases out there, but they're very rare, especially in the United States. So this is not a big deal in the United States. It's a bigger deal with the ESBL. It only detects CTXM. And in the United States today, that is about 95% of ESBLs. But that leaves 5% of your ESBLs that are now TEM class or SHV classes. Those are the original classes of ESBLs. It cannot detect those. So just because that CTXM is not detected does not necessarily mean that there's no ESBL there. And we have certainly seen that. It detects the presence of the gene, not the actual expression of the enzyme. So that could be a bad thing. For example, we relatively frequently get a bug called Acinetobacter radioresistance in blood cultures. So it detects Acinetobacter. It detects an oxygen, but the oxygen in this species is defective. It is not expressed. So the bug ends up being completely resistant. So when we see this, we call our pharmacy. We say, we have an Acinetobacter with an oxygen, and they're thinking, oh, great, this is going to be a carbapenem-resistant Acinetobacter. And they treat for a couple of days as if it's a carbapenem-resistant Acinetobacter. And then after we get our susceptibility back, we say, false alarm, it's susceptible to everything. On the other hand, it can be a good thing. For example, I talked about different levels of expression. We have gotten 2 blood cultures with E. coli where it detected a KPC gene. And then when we did susceptibility, it was totally susceptible to the carbapenems. How did that happen? Well, that's because only about 1 in every 10,000 cells was actually expressing the KPC. So when we put it on our susceptibility instrument, it looked like it was susceptible. But on the other hand, if we had treated that patient with a carbapenem, then we may very well have selected for that 1 out of 10,000 cells that was actually producing the carbapenemase, and treatment would have failed. So it's great, it's fantastic, but there are some little quirks with it that we have to be aware of, especially with those gram-negatives. So the next step then is going to be, how do you go about treating these? And I'm not going to talk about that. Here's what we do. We grow it up. Actually, the next step is we grow it up on plates, we identify it with the MALDI, and we do susceptibility testing. And then we're going to talk about treatment. And I'm going to leave that for next week for Matt to do for us. That is all I've got. I'm happy to entertain any questions if somebody's got some.
All right, really excellent presentation, Dr. Junkins. Thank you so much. And I welcome everybody back for part 2 of this presentation. Thank you.